What if you could tap into the body's earliest developmental blueprint and harness it to reset our tissues, rejuvenate aging organs, and truly extend health span? Today's guest is doing exactly that. They're working at the cutting edge of stem cell science, unlocking a new standard for regenerative medicine. Hey everyone, welcome back to Decoding Longevity. I'm your host, Raghav Sehgal, joined with my co host, Macsue Jacques.
Hey everyone. In our last episode, we have explored a potential new drug discovery that has a really incredible potential to atherosclerotic plaques forever. So it's really, really exciting times. And today we're taking even a step further into the future, exploring how we can reset aging altogether. So our guest today is Yuta Lee, the co-founder and CEO of Accelerated Bio, a company pioneering the use of stem cell technology to rejuvenate tissues and extend health span. Yuta, welcome to the podcast. Thanks for being here.
Yeah, thanks, Macsue, Raghav. It's really, really good to be here.
Yuta, so Accelerated Bio work sits at a really interesting intersection of longevity science and regenerative medicine. For listeners who might not be familiar, could you explain what the company actually does and how do sort of stem cells fit into this picture?
Well, Accelerated Bio is working on the human trophoblast stem cell, or the HTSC. These happen to be the earliest stem cells that you can source without any ethical issues. Now, before I jump into how we source the cells, I think it's really important to kind of frame it by giving us a mental map of where all the stem cells come from in the first place, right? So in the context of fetal development, the first 8 weeks is the embryonic stage. And then from 8 weeks to the 38th week is the fetal stage, right? That's when our organs are developing, our body parts are developing. And then you have the birth event at 38 weeks. And then everything after the 38 weeks is considered adult, right? And as a rule, the earlier you go, the more biologically active the cells tend to be. Now, the earliest stem cells that you could source is the human embryonic stem cell, or HESC. These are typically leftover embryos from IVF clinics, typically at like 5 days post-fertilization. Now, if you use these cells, it causes a lot of ruffle because it's some people consider it an ethical issue, right? Because you're destroying a potential life, right? But that is the earliest cells. Then the next is ours, the human trophoblast stem cells, or HTSCs. We source our cells from between 4 to 8 weeks post-fertilization from an ectopic pregnancy. Okay. Now, my father actually discovered this platform back in 2003. My father is Professor Jiaonan Li. He's an MD PhD. He received his MD from Tohoku University in Sendai, Japan, and his PhD from St. Bart's University of London. And so in 2003, he was performing a procedure called a salpingectomy to remove an ectopic pregnancy mass when he discovered this HTSC platform. So an ectopic pregnancy occurs when, at the early stage of pregnancy, when the embryo is traveling down the fallopian tube, sometimes it will get stuck. And if it continues to grow there, it'll burst the tube. And if it bursts the tube, it might cause internal bleeding and the mother may die. Okay. And so what typically happens is a woman will feel an excruciating pain, go to the ER, get scanned, and the doctor will say, well, you have a tubal ectopic, we have to remove it right away. And they have to schedule an emergency salpingectomy. And what they do is they ablate both sides of the tube and they use a scope to remove the mass. And once they do, all they have to do is give it to the pathologist to double check it. Yep, it's an ectopic, and they can throw it away. Okay. So my father in 2003 was performing this procedure, took the mass out, and was thinking to himself, why are we throwing this stuff away? There's got to be amazing stem cells in here, right? But to avoid all the ethical issues, he scrapes off the preplacental tissue off the top of the embryo, hands the embryo back to the pathologist who checks it and throws it away. So we've effectively sourced preplacental tissue from the embryonic stage without any ethical issues. We've been working at it with the stem cells since 2003. We've already taken one line all the way to GMP, and we're using it as a novel stem cell source for regenerative medicine. So I think that, in relation to that, after our cells, you can get cells also from the fetal period. But unfortunately, most of those cells are from fetal abortion. So that's kind of sticky. No one wants to use that. And so the most cells you can get is from the birth event. So the birth event, you can get a lot of cells, the placenta, Wharton's jelly, cord blood, amniotic fluid, for example. And then everything after that is adult, so you can get cells from adipose or fat or bone marrow. And the very last part is the iPSCs that you may have heard of, induced pluripotent stem cells from Shinya Yamanaka, right? So he used a virus to load 4 transcription factors and was able to turn a stem cell from an adult cell all the way back to a stem cell state. I believe that's arguably the biggest milestone that we have in longevity, right? So the idea is that if you're able to change or reprogram one cell all the way back to the stem cell state, the earliest state, that means it's theoretically possible to change every cell in your body back to an earlier state, right? And so that makes longevity not science fiction anymore. It really is just an engineering problem that we all need to solve.
Yeah. Look, I seen your talk, I think last year for the first time, and I was super taken by it, how you actually changing something that is so painful and so scary for mothers and actually turning into something that can be so good and help so many people. So full credit to you for and your dad for doing this. It's quite incredible. I thought it was amazing. Anyway, so that sort of like brings a little bit towards the next question. So about safety. When we talk about reprogramming cells, like as you just mentioned at the end with the OSK, and then now there is new research coming showing that there is like a single factor that can rejuvenate the cells without really going into that pluripotent state. So I guess like there's always the risk of pushing too far or like leading to that loss or uncontrolled growth or cancer. How is your company specifically addressing that challenge?
That's a great question. Our human trophoblast stem cells actually are primary cells that require no reprogramming. Right? So we source them directly from the preplacental tissue. And so these cells are naturally senescing cells. So after a while, they experience cell cycle arrest and they just stop growing. Okay. But luckily, because they're such early cells, we're able to drive them out to 85 population doublings. Now, population doublings, as you guys know, is 1 to 2 cells, 2 to 4, 4 to 8, et cetera, 85 times. And that is actually enough cells to grow 2 trillion human beings. It's e to the 25 cells. If you can imagine 1 with 25 zeros behind it, right? So scale solved. And with that kind of scale, we're able to do a heck of a lot. The second most important characteristic that we have is the HLA-G. So these cells are from a preplacental source. Now imagine this. The embryo itself is a recombination of the father's gene and the mother's gene, right? So technically it's an alien thing to the mother. So how come her immune system doesn't try and inject it? Well, it turns out the placenta has a very special role to dampen down the mother's immune system so that the embryo can stay attached. And it's with this marker called HLA-G. And our cells, because they're preplacental in nature, express this. And even when we differentiate them toward neuroprogenitors or pancreatic progenitors, they stay. So these cells are really quite unusual and probably the most ideal allogeneic cell source they could possibly use. So we have the combination of scale and immune privilege at the same time. So there's no reprogramming like with a lot of the immortal cell lines, right? The immortal cell lines are cells that continue to grow and they don't ever stop. These are the human embryonic stem cells and iPSCs. So relative to them, we don't have that risk.
Yeah, that's really cool. And in some ways, right, the HTSCs are the most natural form of cellular rejuvenation, one can argue.
That's right. That's right.
It's the natural form of occurrence of cellular rejuvenation, right?
That's right.
How humans for generations have been having cellular rejuvenation.
That's right.
Which is what I love about this approach in some ways. So I guess in some ways that answers the next question I have, but I want to ask from your perspective as well. So in this context, these early HTSCs, what exactly does cellular rejuvenation really mean to you? Right. And could you walk us through how your platform actually does this, right? Are you working with sort of specific tissues or targeting sort of this general rejuvenation pathways?
Right. I have to admit, I didn't come up with the idea. So what happened was about 2.5 years ago, a researcher from the National Institute on Aging calls me up And he goes, Utah, I saw one of your presentations in Seattle, and I hear you have the earliest stem cells without any ethical issues. I said, yeah, that's what we have. In fact, we're taking one of the donor lines all the way to GMP. And he asked me for all of my conditioned media, right? That's the media, the soup, the nutrient-rich soup that you grow cells in, right? And normally you throw that away, but we actually kept everything. So I sent him 6 liters of frozen media. Then I called him up and I said, hey, man, what are you doing with this stuff? And that was the first time somebody ever told me about the heterochronic parabiosis studies done about 20 years ago at Stanford with Irina and Michael Comboy. And that's when they stitched the 2 mice together to one circulatory blood system. And they realized that the old mouse was getting biologically younger and the young mouse is biologically getting older. The second experiment they did was they did not do the stitching, but they did a plasma transfer and the same thing was happening. So what did they discover? Is that the rejuvenation of an old mouse is really just a function of the proteins coming off the blood of the young mouse, right? And that's why you have all these tech billionaires in Silicon Valley trying to get, keep blood boys and donate plasma to them to stay young. So the researcher came and said, hey, you know what? If you can rejuvenate an old mouse just by the protein of the blood of the young mouse. I wonder what would happen if you were to take the earliest stem cells and take that secretome, the proteins coming off of that, and put it towards senescent cells. And that's what they've been working on for the last year and a half. And that data came out pretty fantastic. And so what we discovered is that in vitro and in vivo, we were able to systemically downregulate all of the SAPs, which is the senescence-associated secretory phenotypes, all the inflammatory messages that senescent cells are putting out. And so based on that, we started looking into indications that we can go after. And so we are actually not using the cells themselves, but we're using the exosomes or the extracellular vesicles coming off of the cells as a way to address senescent cells. So that's the first thing we're going to do. And then after that, we will approach longevity as a long-term vision.
Yeah. That is super interesting. And I also just have another idea. Imagine if you get those old cells and you put in those juices where the young cells are actually, secreting those things. Will those cells then potentially become a little bit younger? That's that would be super
right.
Cool. And yeah, and the exosomes as well. It's such a new thing that has been studied. We've been talking about it a lot, but no one has really gone deep inside and broken down what is and we keep finding out more and more that there's more things inside than we just thought previously, which was waste, but the initial idea.
That's
so
right.
It's very
That's right.
exciting. Many breakthroughs in this field still happening in the lab. There is new things that are being discovered every day. So it's really exciting. But Accelerated Bio seems to be pushing towards real world applications. Could you potentially, I know that you mentioned some, but what are the areas where you see the first clinical or therapies being used or emerging from those sort of researches or from yourselves?
That's right. So because of the NIA data, actually 2 doctors from a very large hospital group, which I can't name yet because we're negotiating with them, approached me and said, hey, we actually have over 800 long COVID patients that we don't know what to do with. Right. So there are 5 different indications that are very similar. These are all what they call post-viral infection, low chronic inflammation diseases. And they are wreaking havoc over a large population of people that got COVID or other diseases like chronic fatigue syndrome. And so we are actively working with them to head towards IND in phase 1 for one of these indications, most likely fibromyalgia. And so that's our first indication. The thing is, we really believe that because of the systemic downregulation of all inflammation, we can apply the same material to anything that is inflammation based, right? So you can talk about rheumatoid arthritis, IBD, lupus. These are all inflammation-based diseases, autoimmune diseases, right? We just don't have the domain knowledge for these. And so if there are anybody in the audience that has a company or a research program working on some of these inflammation-based diseases, please come talk to me, because we're like a big hammer looking for nails to hit. We kind of see ourselves as like the NVIDIA chip for the industry, right? We have the earliest cells. We have scale and immune privilege. We have all the IP around it. And the only thing we lack are domain knowledge over indications and patient access. So if you happen to have an idea to say, hey, I want to start a company, to go do this, guess what? I already have the material that you can start with. And what better thing to start with than a stem cell platform that is ideal, that has all the IP around it and already manufactured GMP. So if you guys have any ideas, call me.
You, you heard it here first.
That's right.
Gotta start a new spin-out company in the inflammation arena. Gotta
That's
come talk
right.
to you, Yuta.
That's right.
He's doing some really cool work. But one other cool topic I want to talk to you about, Yuta,
Yeah.
is how you're quantifying some of these things, right? Using
Yeah.
some of the really cool biological aging metrics that you're using in your
That's right.
experiments. So can you talk about how you're using these biological aging metrics and tools to sort of track the effects of
Right.
rejuvenation?
Yeah, that's great. Great question. Which I have a question for you guys. I mean, for me, we've been looking into the extracellular vesicles or the contents of the EVs through multi-omics, right, to evaluate the SASP readings, the inflammatory markers, right, in vitro and in vivo. And we're actively looking into biological clocks now because we want to try and use one together to go into clinical trials as a potential tool for human trials. Now, my issue is there's so many clocks out there, right? And I'm not completely up to date on all of them, but I know you guys are. So my actual question to you is, given that I don't know how many clocks are out there, like 50, 100?
Oh, probably more,
Insane.
way more. So
Okay,
many,
all right, like hundreds of clocks, right?
hundreds
So
of them, yeah.
okay, so my question to each of you, obviously I want you to answer separately, is do you think that we are even close to being able to identify or develop a one true biological clock, a clock that rules them all? Is that possible?
It
Maxwell,
feels
you
like
want
Lord of the
to,
Rings.
yeah, Maxwell, you want
That's exactly
to,
my reference.
yeah.
It feels like Lord of the Rings, a clock, or a ring to rule them all. I guess
Right.
I don't think a clock in itself will be the answer, but rather an integration. And I
Right.
think we're starting to tease out some of the things when we talk about foundational models and we talk about digital twins, because I think all of those omics and methylation proteomics, they are all interlinked. And if you're measuring a clock using proteomics, or if you're measuring clock using DNA methylation, you're gonna get different answers. But if you think that
Right.
everything feedbacks itself, we're only gonna have something as such that is gonna be when you integrate everything. And when you have a foundation model
Mhm.
that potentially reflects a digital twin and gives that sort of like idea.
Right.
I guess
Yeah.
I think that's my take. It's going to be much more personalized than generalized. It has to
Yeah.
be for
I think,
each person.
yeah.
What do
I
you
think my
think, Raghav?
I think my take is I
Yeah.
think there's not going to be one clock to rule them all. Of
Okay.
course. Well, my take is probably we will have clocks for specific purposes.
Okay.
What I mean is there'll be a clock that'll be really good at measuring inflammation, right?
Mm-hmm. Mm-hmm.
So we have like I don't know if you were you at the biomarkers of aging conference?
Yes, I
You were.
was. Yes.
Yeah, so
Yeah.
there was this whole talk, Jamie Justice's session about like
That's right.
the image inflammation and immune clock, right?
Right.
Which is about specifically measuring immune aging really
Mm-hmm.
well, right? Or the IH clock that was developed by David Furman. So
Right.
it's a very, very specific clock doing a very, very specific job, right?
I
And I think we'll have clocks that do very specific roles, very specific, measuring very specific dimensions of aging
Mhm.
very
right.
um and and these global clocks are
Yeah.
good to a certain
Right.
extent in
Right.
capturing sort of global behaviors, but to really capture nuances and sort of nuanced effects, which is what we'll often require with these interventions, we will require sort of these more specific clocks. I don't think sort of these one clock to rule them all might be.
Yeah, I think Andrea also put it very beautifully at the conference, where she's actually using all of those different clocks to measure and see which conditions they better reflect and they give a better
Hmm.
answer for.
Hmm.
So I think that's a beautiful
Okay.
way where you can combine all of that and then just sort of like compare and come up with a
Yeah.
guideline and say, okay, are you trying to
Right.
measure cardiovascular health? Use XYZ
Right.
clock. So I guess it would
Right.
really depend on the question itself. And that's where ensemble clocks that was done
Yeah.
recently in mice. Once that is
Yeah.
translated to humans, I think that will be great because it will combine all of those different interventions into a one thing where you can just go to that and
Hmm.
say, okay, which one is the best, and sort of like auto selects based on all this plethora of clocks that have been developed. So
Yeah. Yeah.
That's
yeah.
right. That's right.
It's
Yeah.
definitely.
Yeah, I was just using this as I was asking a question based on the model from the financial industry, right? So you
Yeah.
have
Mm.
a very large stock market, but then
Mhm.
with different sectors, biotech, finance,
Mm,
high tech, but then you have an S&P 500 that
mm,
tracks the index, which is like an index that tracks all of them across the board,
yeah.
right? And
Yeah.
so I think I'm just thinking from a very general consumer standpoint, how great would it
Mm.
be to have something like that for the biological clock to kind of
Yeah.
just
Mm.
give somebody an indication that without a scientific background or having to go
Mm.
get a PhD like you guys to really understand what that means. And so I don't know, I'm hoping, I'm being very hopeful and positive, that
Yeah.
optimistic, that hopefully you guys could create something that will rule them all.
Yeah. Well,
Yeah.
maybe we can just have a platform where you host everything and people can just go in and it auto selects and all of that. So you're not eliminating things, but you actually
Right.
putting all under one umbrella, so you don't have to go to 10 or 50 different places to measure
Yeah.
There you
everything.
go.
Yeah.
That's right.
Yeah,
That's right.
that's an idea.
Yeah. Okay, so moving on to our next
Yeah.
question, I think looking a little forward and looking into the future a little bit, where do you
Yeah.
see this field sort of evolving into in the next 5 years? Will cellular rejuvenation really sort of stake its claim and become part of sort of the mainstream longevity therapies? Or do you still think it will be more experimental for now in
Yeah.
sort of the next 5 years?
Yeah. That's a great question. I think that I'm really excited about what's happening right now because I believe that we're really at the cusp of scalable rejuvenation, right? So there's
Mm.
so many different companies working on so many different strategies. I think the most exciting one for me, other than what we're doing, is the partial reprogramming, right? So there are companies like Retro or Life Biosciences that are doing amazing work with partial reprogramming, and I think it's really promising. From us, we're using at Accelerated Bio, we're using stem cells. Our approach is stem cell therapy and the exosome, right, to systemically downregulate inflammation. But that's the thing about the longevity industry in general that I love, is that we don't really see each other as competition, right? I mean, there's so many aging related diseases that we need to solve, and everybody should just jump in and try. And if you don't have a strategy, at least you can try and invest something in longevity, right? Because especially if you're over 50 like me and you're ultra high net worth, I mean, what does beating the S&P 500 for another 12, 15 years mean to you? Zero. You can't take it with you anyway, right? So everybody should be working on longevity, right? And so I think that beyond the science, the next big catalyst probably has to be the regulatory landscape, right? So I want to direct everybody's attention to the fact that there are states out there like Utah, Montana, New Hampshire, and Florida that are they've already passed right to try laws. So
Yeah.
that really just allow terminally ill patients to access advanced therapies as long
Yeah.
as they pass phase 1 for safety with the FDA, right? And I think that really helps with shortening the access to these therapies.
Yeah.
Now, I believe that once that becomes a success, they'll start expanding into more even earlier ways to access this stuff. But I mean, I think based on that, I really do believe that longevity is lives in prevention, right? And
Yeah.
so, as Brian Kennedy from University of Singapore always says, and I'm paraphrasing, it's much easier to maintain homeostasis than try to fix something when it's
Mm,
gone completely wrong. Unfortunately, the current FDA is set up for sick care, but imagine if the FDA could set up a preventative section, like a section before all the sick care, right? That
mm,
means that a lot of this preventive therapies can be put there and regulated, so you can eliminate a lot of the non-scientific snake oil people, and really have a proper pathway for VCs and other institutions to invest in preventive care. And I think that's going to be far more cost effective than over time than we currently have. I think these two aspects are really exciting for the future: the science and the potential change to regulatory environment.
Yeah. Absolutely. This is something we always touching on, and always going on and on and on, like how can we shift that to be more preventative. Almost kind of like a broken record, but it's true, and hopefully it gets through.
It's true.
Anyway, before we wrap up, I think I have one final question for you. What is the thing that you think is the most exciting right now, whether it's like a new discovery, a new collaboration for you, or a future goal personally or for Accelerated Bio to achieve in the short time?
As Aubrey de Grey points out, we're very, very close to longevity escape velocity. I think that excites me the most. And I agree with him. I think within the next 15 years, I hope that we can develop a lot of these technologies and reach the clinic and really allow us to live healthily beyond 150 and maybe even achieve immortality. And I'm excited that Accelerated Bio is taking part and partially in that goal. Right. And I really wish that for our children, actually. Think about this. Like, the last generation grew up with the internet. This generation is going to grow up with AI, right? And the next generation is going to grow up without any anxiety for death. Beautiful. I mean, it's truly amazing how much can change in 100 years, right? It's incredible. And so we live in interesting times. So live it up and be healthy. First rule in longevity: don't get in a car accident.
Yuta, I'm stealing that line. I love it. The next generation will grow up without having the anxiety of death. Oof. That hits
Take
so
it.
Yeah, that
Take
gave
it.
me goosebumps.
Take it. It's
Yeah,
it's really
definitely.
great.
Yes. Yeah. Love it.
Yeah. Yuta, thank you so much for joining us today and for sharing your insights and the incredible work you're doing. And the conversation was really, really insightful, and how highlights really how things that sometimes can go wrong can be turned for the good. And I love that. So this is just not a tool for replacing damaged tissue, but it really could be reversing the whole aging itself. So well done, and thank you for doing this work.
Yeah. And to our listeners, we link Accelerated Bio's website and research updates in the show notes. If you enjoyed this episode, don't forget to follow, share, and leave us a review. Thanks for tuning in, and we'll see you next time on Decoding Longevity. And once again, thanks, Yuta, for joining us.
Thanks, everybody. Thanks, guys.
Thanks.